We'll go to our next presenter, Dr. Christopher Lindsell. So I'll give the introduction. He's a professor and co-chief of biostatistics at Duke University. He's the director of data science and biostatistics at Duke Clinical Research Institute and serves as the multiple PI of Vanderbilt Trial Innovation Center. He's also editor-in-chief of the Journal of Clinical and Translational Science. His research interest is in application of rigorous data generation and analysis methods in acute care environment and intersection between emergency care and public health. He's led multiple data coordinating centers for multiple multi-center clinical trials, including different types of trials such as network, pragmatic, embedded trials, and many others. So thank you for being here, and we're looking forward to the talk.
Many thanks indeed for a very kind introduction, and I'm glad to be here. Welcome to a slightly different talk. I guarantee that there will be few equations, and those equations that I do include are mostly words as opposed to symbols. And this is very purposeful because this conversation, this last session, is really intended to help you think a little bit more about designing for diversity as opposed to kind of analyzing for diversity. So this is a question, a conversation around design, and it's a practical conversation. So we'll start out by talking a little bit about design for diversity and how it helps to prevent health disparities. We'll talk briefly about guidance that exists. We'll talk a little bit about some of the design approaches. And at the very end, I'll just make a plea for systemic change that help us to reduce health disparities through proper design for diversity. A few disclosures: I do receive funding from NIH, some foundations, and some pharmaceutical companies. I do consult with a couple of companies, and I sit on data and safety monitoring boards. As Gayatri mentioned, I'm the editor-in-chief of JCTS. I normally have to acknowledge GPT and Copilot these days. I'm glad to say that this particular presentation is all my own thinking. So if there is anything incorrect or something you would like to disagree with, please let's have a conversation. We do not need to blame our artificial intelligence. It's human failure, not artificial. So the first question is why? Why do we want to do this? Well, to begin with, health disparities, we'll just consider the concept of health disparities. These are factors that contribute to differences in health statuses and outcomes among people. They can be environmental, sociocultural, behavioral, biologic. A health disparity, very importantly, it is a preventable difference in health status and outcomes. Generally adversely affects populations. We all have experiences with factors that contribute to disparities. We know exposure to stress, whether it is racism, whether it is sexism, whether it is discrimination against ableism, whether it is any form of stress and discrimination, can result in both biologic change and challenges in engaging in the research process. The impact of lifespan cannot be underestimated. Work exposures, the impact of being a working mother on ability to engage in clinical research is a really important thing to think about. As people get older, the consideration of comorbidities, and as we design studies that exclude people with comorbidities, we're kind of adding to an exclusion criteria that is not comorbidity, it's actually age. And does that lead to a disparity? If we require participants to come to our medical center and travel to our medical center, are we excluding participants accidentally who don't have the functional ability to get to the medical center? Similarly, when we have inclusion criteria related to mental health, are we accidentally including people from our studies inappropriately? And when we don't consider social drivers of health, we may have even more concerns. So when we do not consider factors that change outcomes for some people differently to others, research can contribute to a preventable difference in health status and outcomes. This is a critical statement, and I posit that to this point in time, clinical research has contributed to health disparities by failing to design for diversity and failing to acknowledge factors that contribute to understanding what changes outcomes for some people and not for others. A flawed evidence generation system compounds health disparities. If our evidence is systematically biased, how can we truly impact health? If the evidence systematically excludes at-risk populations, how are we truly helping people? And if our evidence ignores the complete contribution of lived context to outcomes, how on earth are we going to change outcomes? Now, there is a popular solution to this whole let's design for diversity and understand health disparities and fix it. And that is, let's measure and adjust for diversity variables. My favorite topic, let's measure and adjust. So I'm just going to kind of think through two kinds of studies, clinical trials and observational studies, fairly similar. We start out at the beginning with kind of a purpose, whether it's to compare interventions to draw conclusions about effect, or whether it's to assess factors associated with outcomes. The rest of it is fairly straightforward. It's you find people, enroll people, randomize people, do the intervention or measure what intervention was done. You collect the data, and then you compare groups. It's a fairly standard approach. And when we adjust for diversity, we start thinking about health disparities at the end of the study. We start thinking about it during the compare groups phase. The challenge is we frequently get the variable wrong, or we use it incorrectly. And I would posit that when we design for diversity and we think about diversity at every single point in this process, we would be able to better draw meaningful conclusions relevant to all populations without having to do statistical manipulations on the back end with mistakes and accidents. So design for diversity is prevention. By designing for diversity, we can begin to address the problem that our evidence generation system does not actually result in generalizable information. We can begin to develop an understanding of factors that contribute to the success or failure of interventions among diverse populations. And we can remove the evidence generation system as a contributor to health disparities. And I would point out that in today's research environment, design or diversity is not a choice. NIH requires targeted and planned enrollment tables to really try to help the investigator to think about, can my population reflect the at-risk population? Now, whether it truly works, I'm not certain, but it's an intention. Similarly, we have FDA guidance with the intention to improve the diversity of our patient populations. And moreover, there is a lot of social desirability, particularly among our patients and participants, to ensure that we design relevant for people. If we are designing relevant for researchers, are we answering the proper problems? So let's think a little bit about the guidance. This is the help that exists, and it gives us some direction. The FDA has recently issued formal guidance, and it's intended to ensure that there is enough evidence to generalize from your studies to the at-risk population. And the intent is to prevent systematic errors. The classic example is pulse oximetry, but there are many other ways where failure to design for diversity can result in systematic errors. Using a race-corrected EGFR as an inclusion criteria could bake in a systematic error if it is not properly considered. So early in the development process, no later than the end of phase 2, the guidance really recommends, submit the development plan, make sure that you are designing for diversity. This is for all kinds of studies, drugs, biologics, devices. And the FDA does expect to see a diversity plan. It goes beyond reporting on subgroups or simply analyzing as a subgroup. It forces and really recommends that we assess the diversity variable. We consider how we will analyze it. We implement operational plans to recruit and retain a representative cohort within which that diversity variable is meaningful and can be analyzed. And at the end of the day, we want to generate evidence when there is an expected impact of the diversity variable. This is particularly important. In our studies, if we think there may be an impact of a diversity variable, there's a plausible mechanism, we absolutely must be designing to include that within our studies and understand it. If there is no known mechanisms, then we do not understand the contributors to disparities, and so we don't know which diversity variables to focus on, then the guidance just suggests use the demographics or the characteristics of the at-risk population. There are some instances where investigators will lean on the demographics of a geographic region. I see many times, our population looks like the US population. Frankly, a disease at-risk population very rarely looks like a geography. And so perhaps don't use those demographics, use something more explicit. But ultimately, this is not that hard. It's very explicit. Consider the cause of the disparities, consider the diversity that matters, make sure you design this into your study intentionally and not accidentally through adjustment at the end. So the guidance does start to point us in the right direction. However, the guidance, in my opinion, and this is my personal opinion, and this is showing my heart on my sleeve and my advocacy here, we have to go beyond the traditional race, ethnicity, and gender issues. We have to worry about ableism. We have to worry about being able to include people with different languages. People can have the same race, ethnicity, and gender, but have a disparity in access to care because of a language difference. And so we need to go beyond, and we need to think about the causes of disparities and ensure that when we design for diversity, we bring all of those issues into what we're doing. I would also posit we need to scrape beyond the surface. We have to ensure diversity. We can't just say we opened the doors to everyone and we got what we got. The only time I consider that even a marginal excuse is when you've got an emergency department or an ICU study and you do have what you have. But even there, you can choose your ERs and ICUs to be located in areas where the catchment population is more diverse. So we need to start thinking through things like compensation, language assistance, the location of our sites, how we transport people, or how we move studies to people, how we engage the communities and how we minimize burden on people. Indeed, I really strongly advocate for involving participants in the design and conduct of the study. That is a surefire way to improve design for diversity. And this all, of course, leans on top of rigorous measurement and analysis. So I think design for diversity is an optimization problem. That's the way that I tend to look at it. Historically, as we design a clinical trial, we optimize to have objective measures, rigorous assessments, absolute control, total constraints, and we obtain as much information about the disease, the mechanism of disease, and we future-proof everything by measuring everything and taking all kinds of specimens. If we think differently, and instead of optimizing for our own beliefs and our own needs, and we optimize for the participant, we can still ensure objective measurement with rigorous assessment. We control the intervention. But instead of constrained processes, build in flexibility that allow us to increase the diversity of our populations. Focus on obtaining the information needed to answer the question, the primary question in the study, and to inform the disparities or the diversity question. But all of the extra work is an impediment to engagement in research because the extra information is irrelevant to the participants who want to engage. The more that you add irrelevance and the more that you add burden to participants, the greater you are going to result in a non-representative, non-diverse patient population. So historically, we really optimized to control the system. And design for diversity, I posit we should optimize, not spending money to control the system, but to make the system generalizable. It's a different problem. Don't optimize for the researcher. Optimize for the participant. Now, it's actually easy to optimize for the researcher. You know, we're all researchers. We know how to do this. We've done it forever. I've done 30 years of research. I know how to make my trials function. Optimizing for the participant is hard because we are not the participant. Now, I say that sometimes and people look at me and go, you know, I'm a patient too. But we are still researchers. We still understand how the sausage is made. And so even though we understand how the sausage is made, it changes how we might think about the research process. And it means that we are not necessarily optimizing for a research naive participant. And that becomes critical. A slight aside here. As we think about design and design for diversity, I forgot to mention this earlier. As we think about clinical trials and observational studies, they can be much more similar than we think. Instead of the observational study to being assessed factors associated with outcomes and to draw conclusions about their likely effect, if we design our observational study as though we were randomizing people to the exposure of interest, we can actually enhance or improve the lack of bias in our causal effect. And this kind of an approach, if built in correctly at the design phase, we can include the diversity or disparity variables in the design for randomization. I'm not going to talk about the process, the approach, the methodology. There are lots of papers out there that do it. If you see the words target trial, trial emulation, simulated trial, these are things that are approaches that you can get a better causal inference and incorporate a diversity or a disparity variable without adjusting at the end. So in observational studies, we are not stuck with adjusting at the end. We can use some of these tips and tricks to do a trial emulation to include that in the design phase, not the analysis phase. So let's get back to design for diversity. Optimizing for the participant. How do we do that? What is that? Well, I think it's really important that the first thing we recognize is that we need to get out of our comfort zone. We need to listen to and work with participants to understand what works for them. It's really hard. Again, I've been doing this for 30 years. I know what I'm doing. I am wonderful at this. But the reality is, every time I talk to a participant, I realize how little I actually know about how to integrate my trials into their lived context. So when we optimize for the participant, we still focus on rigor, but we need flexibility. We need to focus on safety, but we need to be practical. We need to focus on complete data and accurate data, but we need it to be simple. I would argue that it's actually very possible to design any kind of a trial. People say, you know, I can't do this. I'm doing a clinical trial. The constraints are too big. No, embrace your constraints. Be creative and find ways to solve the problem. By saying I can't do it because the monolithic infrastructures around me are too big is giving in and is being non-creative. And I would argue that we as trialists and designers, this is our duty. On the left, this is where we typically go. Design from where the participant is. And we say, if I was in this study, what would I want it to be? What would help me give you better data? It's very straightforward to do that. But again, we are researchers first. Embedding participants in every part of the research team from the moment of concept all the way through to the reporting and dissemination is incredibly hard, but it is extraordinarily effective at achieving a diverse population and generalizable results. So as we think about it, we have to think about these questions. Where are the patients and potential participants? How do we meet them? What do their daily lives look like? How do we integrate into their daily life? Again, very early on, I mentioned the situation of a working mother. Inviting the working mother to participate in research that has imaging appointments Monday through Friday between 10 and 4 actually is not going to include that person in the study. We need to understand the daily lives of the people we enroll, and we need to fit around them, not them around us. And then beyond the lives of people, what about the interface with the healthcare system? How do we understand that? How do people interface with the healthcare system, and how does that affect both our data and our access to them? How do we best interact with people to tell them about research? A lot of research is site-based, and it is the relationship between an investigator and a participant, or a clinician and a participant, that leads to enrollment. But that doesn't work for a decentralized trial. It doesn't work for a pragmatic study where on-site consent is not part of the approach. So we need to think through, how do we interact with people, tell them about the research? How do we communicate with them in understandable words and language? We as researchers often use phrases and ideas that are fairly complex, and they're not accessible. And on the flip side, sometimes we don't use words that have true meaning and that are useful. There are many examples out there where people truly do understand the use of a word, and by researchers trying to change it, we add confusion. An example of that is the word randomization. Most people understand what randomization is. They may not understand how or what, and some people are confused and they don't quite get it. But if we get to really, really, really complicated descriptions of what randomization is, participants are like, I'm out, I don't know what this is. And so how do we communicate? How do we go beyond the initial step of meeting people where they are and bringing research into their lives, not the other way around? I will note here for retrospective studies, we can't ignore this. Just because the data already exist, we actually need to understand how those patients end up in our data set. What is the information content of their data? And as we kind of thought about earlier with the concept of a target trial, that's when we can start thinking about that in the design process. So, of course, the solution to all of this is the decentralized clinical trial. And, these kind of a trial, we recruit via social media and the web, great access. We can collect baseline data at a local lab. We can do home visits. We can collect information via the web, use e-consent, grab a central IRB, mail interventions to the patients, follow the patient using wearables, PROs, local labs, maybe just it's an EHR data study. And at the end, maybe we send the participants some compensation for their time and send them the results. And fairly straightforward. I've solved all the problems. I've put my study inside the patient's life, and I've designed for diversity. Well, actually, it's a myth. By making everything virtual, all physical barriers to enrollment are removed. It is incorrect. There are many physical barriers that remain. It is not a cheap solution either. A decentralized trial is not about saving money. It's about doing research differently to optimize for the participant instead of for the researcher. It is also a myth that in a decentralized trial, the data will be poor, the FDA won't accept it, and participants will be harmed. I will promise you a few things. If we do a decentralized trial badly, the data will be poor, the FDA won't accept it, and participants will be harmed. If we just build it for them, they won't show up. We don't actually quite know what we're doing with decentralized trials yet. But if we build it together with participants and we design for diversity from the outset in the DCT framework, we can achieve success on the optimization problem to generate quality information about what treatments work for all people at risk. I just want to share a simple example. Active6 is a decentralized platform trial for testing repurposed medications for acute COVID. It was designed for outpatients with mild to moderate disease, with the outcomes of preventing hospitalization and death and improving time to recovery. It's a very simple study. Patients found out about it online. They read about the medicines. We gave them the information. They consented using e-consent. They chose which of the treatments on the platform they were interested in being randomized to. We randomized them, and then we kind of compensated them at the end after they'd filled out all of their PROs. In this situation, the repurposed medicines were sent to participants in the mail. What were the successes with Active6? So very clearly, we were very able to emphasize the participant or the PRO component, how the participant felt and how the participant symptomatology was exhibited. In the trial, some of our successes were very early on hearing feedback from participants and dropping technology that we thought was facilitative, but was actually getting in the way. As one example, we tried to use a cell phone app to collect data. Participants hated it. They couldn't install it on their cell phones. They much preferred to have a web-based survey that they could pull up on their cell phone that was cell phone enabled. And that was an early learning. If we'd chosen a different app, we may have had more success. But adapting to the participants with their feedback in the process was something we learned we had to do. Minimizing participant burden for data collection was something we were able to do, and it was very important. One of the things we did hear back is actually participants like a regular touch in a trial. They don't like to be left alone completely. And so while we were attempting to minimize all burden, they were actually, hey, still talk to us. We want to be touched. We were successful in enrolling thousands of patients. We've now answered 7 different repurposed drug regimen questions. The 7th protocol was the publication went to medRx this week. We have generated out of Active6 information relative to FDA policymakers and providers. But some of the lessons we learned very early on, we did not achieve diversity because we did not design for diversity. We added in the diversity angle as we were moving forward. By the time we were halfway through the trial, we had achieved a really diverse patient population, but it really required intense effort that if we had the time to design up front, we would have had a more generalizable data set in our early study arms. So that was a lesson that we learned along the way. The second one, the decentralized trial, I mentioned that all physical barriers go away when you go virtual. We discovered there is a massive disparity in the health system, in the mail system. And it's not just rural routes. The mail carriers don't carry mail into inner city areas. They use drop boxes. Mailboxes may go to a Walgreens or a UPS drop space. So inner cities and rural areas are both at risk of not being included in decentralized trials if we rely on the mail system and our traditional mail carriers. So again, thinking about the cause of a disparity and designing for diversity is really important. And then the other major challenge that we discovered here is that even if you include English and Spanish, there are major barriers to access to research that come with having a multilingual society. Whether it is the time-consuming processes for translation and IRB review and approval, whether it is building technology systems that present in multilingual nature, or whether it is ensuring interpretability of language, these challenges need to be brought into the design for diversity conversation. Okay, let's get a bit data sciencey. I've just been talking for a while. I promised I would give you at least one equation. So most people think of data science as using information that exists. I think that the interface between data generation and data use is pretty critical. And so making clinical trial systems that work to bring in the right information is one part of the puzzle. And then making systems that use that information properly is equally critical. So our systems need to have interfaces and language and processes that allow for non-white, non-English speaking, non-male participants to access and engage. This is really, really important because many things in research historically came from a place where the people doing the work had a certain perspective. And that was a long time ago, but the monoliths are taking their time to catch up. We have an opportunity to accelerate that with design for diversity. So let's assume we've solved all of that. Now, what do we do with the data? So in these two cartoons, which one is there a disparity? For which one of these two is there actually a diversity challenge or issue that we really need to incorporate into our design and into our subsequent analytic plan? Does the effect differ based on group membership? Is a different question. It is not equal to the question, what is the effect in the subgroup? Design for diversity in the analytic phase, I would argue, is not subgroup analysis. It's really looking at the interaction terms. And we've heard some really good talks today that have delved a little bit into some of this, some of how you do this. But it's really critical to recognize, does the effect differ based upon group membership is a critical question. What is the effect in the subgroup is irrelevant if the effect doesn't differ between subgroups. Moreover, groups are not binary. Not all groups have meaning. You know, this is the classic. In this particular situation, you've got the y-axis is a treatment effect, the x-axis is age, and you see a point of inflection around the age of 65. This is an incredibly common situation, particularly in health services and health policy research, when we're using retrospective data. And what it comes down to is that break at age 65, it's not really a health thing. It's a Medicare, Medicaid thing. It's an access to insurance. It's a different data stream thing. And so when we're designing our analysis for diversity, we need to make sure that we understand the diversity variable and we pick the right one. You know, for example, with a pulse oximeter, should I measure and assess self-reported race? Or should I evaluate the effect of skin tone on my outcomes? Those are similar but different, and it's important to get it right. Similarly, for understanding the impact of a specific care strategy on outcome, sometimes we see people just add a race variable, and they say, oh, look, race is significant, there's a disparity. You know what? Maybe it had nothing to do with race. It happened to do with perfect confounding by access to care. You know, similarly, I see so many asthma trials where nobody actually measures the environmental exposure of the participant in their home environment. That's not a good thing to do. And maybe that's a cause of a disparity. And a diversity variable in an asthma trial is actually environmental diversity. So design for diversity really means thinking what might perturb the relationship between treatment and outcome, think mechanistically, and then bring those concepts into the design. So central to the guidance that exists is the concept of biological differences, sociocultural differences, behavioral differences, environmental differences. And I would argue that central design should also be reasoned mechanisms. How do I ensure my study cohort represents the diversity of factors that will affect the outcomes given the exposure of interest? And then how do I measure those diversity factors? Now, FDA guidance emphasizes reducing participant burden. In the diversity plan, the who is somewhat pre-specified. However, we have the opportunity to really be creative around the what, when, where, and how of the research we do so that we can ensure a diverse participant population and that we can ensure we understand the diversity issues, the factors that result in different outcomes for different groups. And with that, we can subsequently ensure that our studies are not going to unintentionally contribute to considerable health disparities. So I would argue in this design with the fact that the what is defined, it's we want to enroll a diverse population. The how, what, where, when, the rigor of a data point, it's a trade-off. How hard do we go at that data point? The harder we go at it, the less likely it is to be generalizable. Similarly with recruitment, if we want really easy recruitment for us, it may not be very generalizable. Simple for the research team, again, may not be very generalizable. Cost control may not be very generalizable. I would argue that design for diversity, where we integrate rigor, we make recruitment easy, we make it simple for the participant, and we focus our funds on centering the participant in our design, we actually take the impact of the science up because it becomes generalizable. If we don't design for diversity, the lack of generalizability is an impact, is a negative to our impact. So I would like to leave you with a real challenge. I don't want you to answer these questions today, but I do want us to think about these. Is it possible to truly implement design for diversity without a diverse workforce? So that is just a question that I posit. And then second, how can we as trialists and researchers influence the monolithic infrastructures that for decades have been centered on a set of rules and processes that do not place a diverse participant at the center of what we do? So I challenge you to ask yourselves those two questions as you move forward in clinical trials work. And with that, I will leave you with the statement that in optimizing design for diversity, I believe that we begin to mitigate the historical contribution of clinical research to health disparities. Thank you very much.
Thank you, Chris. Very interesting and thought-provoking. I do have a question for you. Have you seen any published data on willingness to participate in trials? And especially for different diversity groups as you define them, including maybe social class. And then maybe where would, and again, maybe I'm kind of jumping ahead too much here, but some mistrust of research, general research enterprise, does it come into place? Have you seen any published data, is my question.
Yeah, it's a really good question. There are a lot of data out there on willingness to participate, engagement with participants, tactics that help to overcome mistrust. There is information on mistrust. I will share a little bit of data that we generated about 10 years or so ago when I was in Cincinnati. We had a system there where every single patient that presented to the ER was approached by a research coordinator and was asked the question, if during the course of your ED care, you are found to be eligible for a research study, can we approach you? Every single person was asked that question. Then subsequently, if they were found to be eligible, we approached them and we went through the consent process to see whether or not they actually enrolled. And so we have from the moment of presentation through to the presentation of a trial, through to whether or not they consented, the yes or no. Mm-hmm. And we looked at what are the impacts of race and sex, not just of the participant, but also of the research coordinator. And we discovered some really interesting things. For example, we found that women were approached more frequently, but actually said no. And that surprised us because we were of the assumption that there was a disparity in enrollment of women and that they really wanted to be part of research. But at the end of the day, we had a greater decline rate. I posit because of some of the issues I mentioned earlier. The constraints on their lives are much greater than the constraints on the males. We did not find an effect of discordance between race and of coordinator and participant was an impact. Again, that could be because we did not have a diverse workforce. We only had to people that I could include as diverse coordinators. So the data are challenged, but they do exist. And I would encourage us every time we do a study to generate further data as to the barriers to participation and learn how to better design for diversity.
Thank you.